is that, okay, so we had established that the kingdom Animalia consists of eukaryotic multicellular organisms that carry out heterotrophic mode of nutrition. They are also known to exhibit some form of motion, unlike the protozoans that are incapable of moving from one place to, protozoans that are unicellular. The animals are multicellular. Unlike the plants that cannot move from one place to another, the animals can move. All right, and this kingdom Animalia is divided into two subkingdoms, subkingdom parazoa and the subkingdom eumetazoa. So we're discussing now the subkingdom parazoa. All right, the subkingdom parazoa consists of animals that lack specialized tissues and definite symmetry. These organisms lack definite symmetry and they lack specialized tissues. And these organisms belong to two phyla, the phylum Porifera and the phylum Placozoa. Among them, the sponges and the phylum Parazoa are the most well-known. So we're going to discuss about the phylum Parazoa today, the sponges. We're going to discuss about the phylum Parazoa, the sponges. Sorry, the phylum Porifera. We're going to discuss about the phylum Porifera, the sponges today. Now, the phylum Porifera consists of unusual animals that lack definite symmetry or organs. The sponges, they lack definite symmetry or organs. Their bodies cannot be divided into mirror images. That is, if you cut the sponge into two parts, you cannot get two identical halves. And they lack definite tissues. Their cells are not organized into tissues or organs. So they are said to be asymmetrical. But some of them could have a radial symmetry. Some of them very few of them have radial symmetry. Very few of the sponges have radial symmetry. Now, the sponges lack head, appendages, they lack mouth, they lack anus, and the internal organization, the internal structure that is characteristic of animals is not present in these sponges. The sponges are actually the simplest animals. They lack a head, they lack appendages, they lack mouth or anus, and they lack the internal organization, the internal organized structure that is characteristic of other animals. They lack that internal structure. About 7,000 species have been described, and they are usually found in the sea. And they are usually found attached to surfaces in the benthic region. All right, so these are this is an image of a sponge, of sponges. You can see that they do not have a definite symmetry, and their bodies are made up of spores, and that's their bodies are made up of pores, and that's the origin of the word porifera. Porifera. That's why the phylum is referred to as the phylum Porifera, because members of this phylum have bodies that have a lot of pores. Their bodies consist of a lot of pores. And we have seen this video earlier of the various shapes and structures of sponges. They are found attached to substratum in the benthic region. There are various shapes and sizes, and usually they look like plants. The adult sponge doesn't even look like an animal. They usually look like plants. They can't even move from one place to another, unlike other animals that can move from one place to another. So we want to also look at the characteristics of sponges, the characteristics of the organisms in the phylum Porifera. Now, one of the characteristics we want to look at is the habitat of sponges. They are usually found in aquatic habitats, mostly in marine habitats, in the sea. They are usually found in the sea. Just a few of them, some few sponges are found in the freshwater habitat. And they are usually solitary, or some of them could be colonial, and they are sessile. Sessile means usually attached to substratum and not moving from one place to another, and not moving from one place to another. What about their body form? The bodies of sponges are porous because they have so many pores. And their pores are of two types. Sponges have two types of pores. They have inhalent pores, that is, pores that take in water and food into the sponges. And these ones are in the sides or the body of the sponges. And they are called the ostia. Singular of that word is ostium. The ostia are narrow, are minute pores that are on the sides of the sponges. They are inhalent pores. Inhalent pores means water and food go into the sponges through these pores. And they also possess exhalent pores. And the exhalent pores take water and waste products out of the sponge. And those exhalent pores are called the oscula. So the ostia take in things into the sponge, while the oscula take away substances from the sponge. And the oscula is usually bigger than the ostia. The oscula is usually bigger than the ostia. Now here is a diagram of a figure that's showing a typical sponge, the structure of a typical sponge. This is the osculum that takes, this is the exhalent pore where water and waste substances exit from the sponge. Then the ostia, where water and other and food substances go into the sponge at the side. Here, where you have the water flow, this place where water goes in is the oscula. Then you have the ostia, that's where water goes in. Then you have the oscula, where water exits from the sponge. Then, having seen that, we want to make progress to look at other characteristics of the sponges. What about symmetry? Most sponges are asymmetrical. Some have radial symmetry. What about the germ layers, the body layers? Sponges are diploblastic animals. They have just two germ layers, the outer ectoderm and the inner endoderm. They have just two germ layers, embryonic germ layers, the ectoderm and the endoderm. What about level of organization? The fifth characteristic of the sponges, they exhibit cellular level of organization. That's their cells are not organized to perform the same, they cannot work together to carry out specific functions. So they work independently. That's why I would say they exhibit cellular level of organization. Canal system is the sixth characteristic of the sponges. The sponges consist of pores and canals. There are three types of canal systems that are found in sponges. There are three types of canal systems that are found in sponges. We have the asconoid canal system, the syconoid canal system, and we have the leuconoid canal system. These are the three canal systems, sponges, the asconoid, the syconoid, and the leuconoid canal systems. What about the skeletal system? The seventh characteristic of the sponges is that they possess internal skeleton. They possess endoskeleton. And this skeletal system are in between their germ layers. They have internal skeleton. And the internal skeleton is made up of spicules. And the spicules can be calcareous, that is made up of calcium carbonate, or can be siliceous, made up of silicon, or of spongin fibers. They may be made up of spongin fibers, and they are located in the mesoid layer of the sponges. Respiration. Respiration in sponges occurs by diffusion through the plasma membranes of the cells, as it is in protozoans. That's why we say they exist in the cellular level of organization. They do not have respiratory organs. They do not have respiratory system. So diffusion occurs by simple diffusion through the cell membrane, the plasma membrane of the cells. And excretion too, the 11th characteristic, excretion occurs too through diffusion of substances through the plasma membrane of the cells. Okay, so what about reproduction? The sponges reproduce both sexually and asexually. They carry out both asexual and sexual reproduction. Asexual reproduction is by budding and by germ molds. They reproduce asexually through budding or through germ molds. They reproduce mainly through budding or through germ molds. Then we also have that germ molds in freshwater and a few marine sponges. Germ molds or internal boards are formed. Usually, germ molds are internal boards that are formed in some sponges. The sponges are said to be hermaphroditic, having both male and female reproductive organs, structures rather, reproductive structures. And the fertilization is internal. Fertilization in sponges occurs internally. What about the development of sponges? The 13th characteristic. The zygote undergoes holoblastic cleavage. That is a complete division of the zygote. They undergo holoblastic cleavage. And I've said earlier that the larval stage of sponges are usually free swimming. They are usually able to move from one place to another. And that free swimming larva is actually called either the amphiblastula or the parenchyma. And when they are matured, they settle in substrate and they become sessile. So adults are incapable of moving from one place to another, but the larval stages can move from one place to another. The larval stages of sponges can move from one place to another. So sponges are usually found in the bottom of the ocean. They are incapable of moving from one place to another. The sponges, adult sponges, are incapable of moving from one place to another. Now I want to look at classification of sponges. Classification of sponges, that is classification of the organisms in the phylum Porifera. Classification of the organisms in the phylum Porifera. The sponges are classified into 3 classes. And the three classes of sponges include the class calcispongia or calcarea, and we also have the class hyalospongia. The class hyalospongia can also be called hexactinellidea. Then we also have the class dermospongia. So I want to look first at the class calcispongia. Now, as we discuss about these classes, take note of what their spicules are made up of. That's one distinctive feature of the sponges. One thing that distinguishes one sponge, one class of sponge from another is the spicule, what the spicule is made up of. That's one feature that distinguishes them. Now, the calcispongia, just as the name implies, they bear spicules that are made up of calcium carbonate. That's the source of the name calcispongia. Calcispongia. Their spicules, that is the internal skeletal structure, that hard structure that's found inside their body wall. The spicule is made up of calcium carbonate. Their spicules are made up of calcium carbonate. These sponges are verse-shaped. They have a verse-shaped compact structure or loose networks of thin tubes, or they are irregular massive colonies. They can be verse-shaped, or they can be loose networks of thin tubes, or they could be irregular massive colonies. They are mostly small in size. The calcispongia are mostly small in size, and they inhabit shallow waters of all seas. They are small in size, and they inhabit shallow waters of all seas, from the intertidal regions to depths of about 200 meters. The calcispongia are filter feeders. They filter their food. As the current takes food particles and other organic matter, they filter their food. The food enters through the pores and being digested by the food vacuoles. The foods are being digested by the food vacuoles and expelled through the osculum. So the ostium receives food particles, and the osculum, through the osculum, wastes are being passed out. So they are filter feeders. There are about 400 described species in this group. This is an example of an asconoid acerous sponge. It's in the class calcispongia. The name of this sponge is Plathrina plathrus. All right, so it's a member of the class calcispongia. The next class we want to look at is the class hyalospongia. The class hyalospongia. They are also called the glass sponges or the hexactinellids. They are also called the glass sponges. Now, members of this class hyalospongia are basically hexactinal. That's, they possess 6 rays. They are 6 rays, and they possess calcareous spicules. They possess calcareous spicules. Calcareous spicules means spicules that are made up of calcium carbonate. Calcareous spicules means spicules that are made up of calcium carbonate. They possess siliceous spicules. Sorry about that. They possess siliceous spicules. The calcispongia have calcium carbonate. The hyalospongia have siliceous spicules. The siliceous spicules are made up of silicon. The spicules contain silicon. That's why they're called siliceous spicules. They possess silica. And they also lack spongin. These sponges lack spongin fibers. These sponges are hydrophilic. They are water-loving sponges. They are hydrophilic sponges. Their dried up skeletons look like glasses. That's why they are called glass sponges. When their skeleton dries up, it looks like glass. And that's why these sponges are called glass skeletons. Sorry, glass sponges. That's why these sponges are called glass sponges. They are sedentary and range in size from 10 to 30 centimeters. They are sedentary. Sedentary means unmoving. Sedentary or sessile. The word sedentary can also be, the word sessile can be used in place of sedentary. They don't move from one place to another. Their cytoplasm doesn't divide into cells. That's another characteristic of these sponges. They are exclusively marine sponges. These sponges are exclusively marine sponges, commonly fixed firm to hard surfaces. They are usually attached to hard surfaces. Some of them are anchored to soft bottom sediment. They have about 500 species, and examples include the Hexatinella, and other species. Now, this is a typical example of a highly spongy, This species is called the Teller species. Just as you can see, the skeleton made up of silicon looks like glass. So that's why they are called glass sponges. The next class of sponges we want to look at are the sponges in the class Demospongia. The sponges in the class Demospongia. They possess skeleton of either one or four rayed siliceous spicules. So take note, the Demospongia have siliceous spicules. Their spicules or their skeleton is made up of silica, just like the Hyalospongia. So the Hyalospongia and the Demospongia have skeleton or spicules that are made up of silicon or silica. They have siliceous spicules. But the Calcispongia has skeleton that's made up of calcium carbonate. Now, one difference between the Demospongia and the Hyalospongia is that unlike the Hyalospongia that lack spongin fibers, the Demospongia has spongin fibers. They have spongin fibers, or they may have both spongin fibers and siliceous spicules. The skeleton is lacking in a few primitive species. Some of them do not have skeleton. This class have about 4,200 species. This class of sponges have about 4,200 species. They actually are found in the intertidal regions. They're usually found in the intertidal region. Now, one of the families of this class, Demospongia, possess species that inhabit fresh water. And that class, that family is called Spongilidae. So this class is the only class that have members that inhabit freshwater habitats. And the species of this class usually vary in size and form. It can vary greatly in size and form. This class of sponges range from thin excrustations, several centimeters in diameter, to huge cake-shaped species of 2 meters, that's 6.6 feet in diameter. Some sponges are brightly colored, while some of them are not. Now, this is a sponge in the class Demospongia. This is a sponge in the class Demospongia. So that's basically how far we will go today in our lecture. So far, we have discussed about the sponges and their characteristics. In our next lecture, we will discuss the subkingdom Eumetazoa. We'll discuss the subkingdom Eumetazoa. And we'll also look at some phyla in the subkingdom Eumetazoa. All right. So that's basically how far we'll go in today's lecture.